Hello and good day to everyone. So in this particular video lecture, we are going to talk about the different cellular structures and their function. So to have a mental roadmap on what would be the flow of our discussion, let us take a look at the outline. And as you can see, we will begin our discussion with eukaryotes being compartmentalized, which is basically a feature distinguishing them from prokaryotes. And then as we progress, we are going to talk in detail with the different cellular structures such as the cytoplasm, cytoskeleton, the nucleus, the contents of the nucleus, organelles of the endomembrane system, those who are semi-autonomous, and other organelles as well. Now at the end of the video lecture, we are expecting our students to recognize and compare the structural and functional features of plant and animal organelles, and also for you to determine the correlation among members of the endomembrane system. Well, to begin, within eukaryotes, again, they are compartmentalized. They have different compartments, which is a distinguishing feature of eukaryotes, distinguishing them from prokaryotes. Well, by definition, when we say compartmentalize, it literally means to separate into isolated compartments or categories. Therefore, each of these cellular structures is in itself its own compartment, separate from the others. Well, as they have been isolated and separated from other components or from other cellular structures, each created its own function, and that overall increased the efficiency of many subcellular processes by concentrating the required components to a confined space within the cell. Well, like in a factory, each division or unit has its own function or role. So to be able to identify the function and role of the different cellular structures, let us now begin discussing now the cytoplasm and its difference with the term cytosol. So for the term cytoplasm, it is the region between the nucleus and the plasma membrane. So if you focus your eyes in this particular figure, any region between the nucleus and the plasma membrane, that is technically the cytoplasm. It is simply a region. A region wherein it contains different enclosed organelles and other organelles that perform specific functions. The cytoplasm, in addition, is also a site for most metabolic activities of the cell. Again, most metabolic activities of the cell. Why? Because as we take a look at the term cytosol, which is the liquid portion of the cytoplasm, again, the liquid portion of the cytoplasm, one of its most abundant contents would be water, hence a watery substance. Now, what is suspended in this watery substance or in this liquid portion would be proteins, carbohydrates, lipids, and enzymes. Now, given the presence of enzymes, of course, this may tell us that at some point, the cytosol or the cytoplasm as a whole performs some of the metabolic activities. Well, besides enzymes, what is also present within the cytosol are free polyribosomes or free ribosomes in general, further implicating that the cytosol is where some cellular proteins are being synthesized. Proteins such as those that are found within the cytoskeleton. Now, for the cytoskeleton and for our bony skeleton, there seems to be a striking similarity between the two, wherein like our bones, without them, we would not be able to resist gravity enough to walk or even sit up straight. Now, for cytoskeletons, without them, our cells may not resist mechanical stress, an example of which would be the frictional force that our cells are experiencing whenever these clothings are rubbed against our skin. Now, this type of function or this primary responsibility is for your intermediate filaments, a type of cytoskeleton. Hence, as you can see in this particular slide, they provide mechanical strength not only in the cytoplasm but also in the nucleoplasm. So to give you context, what is a nucleoplasm? This is technically the counterpart of the cytoplasm within the nucleus. So it's the region between, or the liquid portion between, the nucleolus and the nuclear membrane or the nuclear envelope. So to give you an idea how intermediate filaments are organized within the cell, let us take a look at this figure. So in this figure, this web-like orange or brown-like structures are your intermediate filaments, found all over the cell, which is quite understandable, of course, as it needs to provide mechanical strength for the whole cell. Now, what is also notable here is that our organelles, such as here, an example, the mitochondrion, does not float, but is anchored to a specific area and are being transported from one area of the cell to another with the help of another component or another type of cytoskeleton, and that would be your microtubules. So in this figure, the microtubules would be the green structures, and as you can see, the mitochondrion is indeed anchored in this particular area, given the presence of this microtubule behind it. Now, one of the last types of cytoskeleton would be actin filaments, which controls the shape and movement of cells. And based on this figure, actin filaments are basically the thinnest out of the three types of cytoskeleton. Now, in addition, they are basically abundant in cells with large-scale movements. An example would be muscle cells, as they are continuously contracting. Now, as you have observed in our discussion of the cytoskeleton, we have already introduced a subcomponent or a subpart of a larger organelle, which is the nucleoplasm, a component of the nucleus. So in this particular slide, it simply shows to you the outside and inside contents only of the nucleus. So you have here the bounds of the nucleus, the nuclear membrane, and distributed throughout the nuclear membrane are nuclear pores. And inside the nucleus are prominent structures that are very essential in information processing, as the nucleus houses the genetic information. So first thing that you need to identify and observe in this particular figure would be this green structure or this green area inside the nucleus, which is basically the nucleoplasm, the fluid-filled part of the nucleus where DNA primarily resides. So DNA resides within the nucleoplasm. However, in this figure, our DNA is in the form of chromatin. So you see, class, DNA in itself has a different form to that of chromatin, but both are genetic information. Both are containing genetic information. So to further understand how they differ in terms of form would be to understand how DNA is being organized. So first thing you need to know is that the length of DNA in most cells, if stretched linearly, it extends to 2 meters, right? A length so great that complex packaging strategies are necessary and are needed to make the DNA fit inside the cells. Thus, we have now 5 different levels of DNA organization only present in eukaryotic cells. So for the first level, we have the DNA or your double helix. So in its helical form. Now, as we progress to the second level, that would be the beads on a string, or also known as nucleosomes. The beads on a string configuration wherein the helical DNA is wrapped around a spool made up of histone proteins. So each of these purple structures are individual histone proteins. So this is one histone protein. This is another histone protein. If bundled together, they create the bead together with the DNA wrapped around it. So you have now the beads on a string configuration. Now eventually, these beads in a string configuration will proceed to the third level, which is the formation of the 30 to 40 nanometer fiber, chromatin fiber, which may be accomplished by twisting the beads on a string structure into a coil. So again, from a beads on a string, you can achieve the third level by twisting the configuration itself into a coil. So at this part, you may now come to understand the difference in terms of form of the DNA itself and the chromatin, as they differ in terms of form and in terms of level as well. So as we proceed to the fourth level, we now have the formation of looped domains, or in technical terms, a 300 nanometer chromatin fiber. So in a sense, this is still chromatin, however, in a different form. Now for loop domains, or for the 300 nanometer fiber, they are formed by having periodic attachments to the protein or RNA scaffold. So like histone proteins, who don't store genetic information, RNA scaffolds don't also store genetic information, but of course without them, the DNA would be tangled and useless. So usually these RNA scaffold are found at the center of your loop domains, in which again, chromatin fibers are having periodic attachments. So yes, they form loop domains for the fourth level. Now for the fifth level, they now form this highly condensed chromosome. So you now know the difference between DNA, the chromatin, and the chromosome. Now specifically for the chromatin, be it the third level or the fourth level chromatin, if compared to your chromosome, of course, they differ in terms of compaction and form. However, besides that, another difference that can be seen between the two would be on what type of cell they are found. Well, in particular for your chromatin, specifically the third level form, in chromatin, right, in third level form, it is found within non-dividing cells. However, as cells progress to its dividing state, given that the cell is indeed continuously dividing, and dividing in the first place may give rise to another type of cell, of course, you may now find chromatin in its fifth form into its chromosome form. Well, of course, during the transition from non-dividing to dividing cell, you will find the fourth level chromatin until it reaches eventually the last or the highly condensed level. So to continue with our discussion, of course, besides the nucleoplasm found inside the nucleus, we also have the nucleolus, which is the site of rRNA synthesis. Well, if you were to ask me what is the function of rRNA, to give you an idea, rRNA is usually paired with certain proteins that make up ribosomes. Ribosomes further classified into free ribosomes or into bound ribosomes. So before we distinguish the difference between the two, you may wonder why ribosomes in this particular slide are termed as polyribosomes. Well, the reason is that as ribosomes are a major component of the translation process, whenever mRNA are being translated into proteins, one must always keep in mind that one ribosome is not enough. Hence, many ribosomes participate in the translation of a single mRNA strand, hence polyribosomes. Because in terms of function, ribosomes may need other ribosomes to completely translate a single mRNA strand. Hence, you have the prefix poly, meaning many. So for a free polyribosome, they are those who are suspended in the cytoplasm. Whereas for bound polyribosomes, these are polyribosomes or ribosomes in general attached to the ER, to the endoplasmic reticulum, making it a rough ER. Hence, you now distinguish one endoplasmic reticulum to another type of endoplasmic reticulum, or it may be bounded to the nuclear envelope. Now, besides rRNA, we also have mRNA, but technically both mRNA and rRNA are both exported from the nucleoplasm to the cytoplasm. But please keep in mind that to be able to export and import molecules into and out of the nucleus, it must be regulated by certain openings known as nucleopores, again located and distributed throughout the nuclear membrane or throughout the nuclear envelope. Now, since the nuclear envelope in this image, as you can see, technically this one, since the nuclear envelope or the nuclear membrane is part of the endomembrane system, then of course we must be introduced to this integrated network or integrated system known as the endomembrane system. So other members of the endomembrane system are the endoplasmic reticulum, be it the rough or smooth ER, we also have the Golgi Complex, we also have the lysosome, the plasma membrane, and of course vesicles connecting the different members of the endomembrane system. So as a whole, they form again an integrated network of membranes and compartments. Well, the organelles of the endomembrane system are related through direct contact or by transfer of membrane segments as vesicles. Hence, what is found in each of the membrane of your endomembrane system is a phospholipid bilayer or a membrane. Hence the term endomembrane system. A system of membranes of phospholipid bilayers within the cell. Hence, you now have the prefix endo, which refers to the term within. So to continue our discussion, let us introduce ourselves with the nuclear envelope. Now, as you can see in this figure, the nucleus is indeed bounded by the nuclear envelope. And so as we enhance and further magnify this figure and proceed to the figure next to it, okay, it is very evident that the nuclear envelope or the nuclear membrane is composed of two membranes. You have the inner and the outer nuclear membranes, separated by a perinuclear space. Now, please keep in mind that each membrane is a phospholipid bilayer. And so as we further magnify this part of our figure, it is now evident that the inner as well as the outer membrane are individual phospholipid bilayers. So to be able to understand the term phospholipid bilayer, you must understand that the main component of any membrane within the cell is the phospholipid. So these phospholipids are those structures dominant within any membrane. So say for instance, in this phospholipid bilayer, this individual unit is one phospholipid. A phospholipid creating one layer. So this is one layer phospholipid. However, opposite to it, you have another layer of phospholipid. Hence, you have two layers of phospholipid, thus the term phospholipid bilayer, which means that you have two layers of phospholipid. Now, a phospholipid bilayer is, in a sense, interpreted as one membrane. Hence, you have the inner and the outer membrane separated by the perinuclear space. Now, one distinctive feature of the nuclear envelope is the presence of specialized channels known as nuclear pores. Well, just to add, nuclear pores are lined and are built by about 30 different proteins known as nucleoporins. But what is important for you to remember is that nuclear pores regulate the entry and exit of molecules between the nucleoplasm and between the cytosol. Now, if you take a look and observe again this part of our figure, it is also evident that at each pore, the inner and the outer membranes of the nuclear envelope are fused together. So they only fuse at sites where nuclear pores are present. Besides nuclear pores, if you take a look at this magnified figure, the outer membrane, to be more specific, is studded with bound ribosomes or bound polyribosomes engaged in protein synthesis, which is similar to the membrane of the rough endoplasmic reticulum. Another feature of the outer nuclear membrane is that it is continuous with the endoplasmic reticulum, making the perinuclear space continuous with the lumen of the endoplasmic reticulum. Now, as we have been introduced to the endoplasmic reticulum, well, morphologically speaking, the rough ER, or the rough endoplasmic reticulum, are usually forming large flattened sheets, whereas its counterpart, the smooth endoplasmic reticulum, are generally forming tubular structures. Well, according to research, this complexity in terms of shape allows for segregation of biochemical processes within the organelle. So this now gives you an idea that both the rough ER and the smooth ER do have different functions. And specifically for the rough endoplasmic reticulum, it is to initially synthesize and sort the proteins, and it performs glycosylation, which in simple terms means the addition of sugar chains, specifically to proteins. Now, the rough ER plays a key role in these functions. And so to further understand this, we need to understand how the RER works. And that we can do by observing this figure in this particular slide. So as the polypeptide, this one, this linear chain of amino acids, is being synthesized by a free polyribosome, of course, by following the instructions of the mRNA, it is now threaded. This polypeptide is now threaded into the cavity of the rough ER. Now, as it enters, the new protein folds into its three-dimensional shape. Also, short chains of sugars are often linked to the polypeptide, making the molecule glycoprotein. Glyco meaning sugar. The addition of sugar chains via glycosylation. Now, when the molecule is ready for export from the ER, it is then packaged in a transport vesicle. Okay, by definition, a transport vesicle is a vesicle that moves from one part of the cell to another, basically a vesicle that transports contents of the ER to the Golgi complex. Eventually, this vesicle buds off from the ER membrane and will proceed to the Golgi complex. However, another type of endoplasmic reticulum is the smooth. But to give you context first on how this free polyribosome was able to attach itself to the rough ER, let us take a look at this image, simply illustrating to you how this free ribosome, or maybe even free polyribosomes in the cytosol, deliver the synthesized proteins or the synthesized amino acid or polypeptide chain into the lumen of the RER. So they simply attach themselves via this complex process. Now for the smooth ER, the following are its functions, but please keep in mind that enzymes of the smooth ER are also important in the synthesis of lipids, including oils, phospholipids, and steroids. Also, when we are in the discussion of the muscular system, we would encounter another function of the smooth ER, which is to store and to release calcium ions, specifically within muscle cells. Now, as products of the ER are packaged into transport vesicles and are distributed and are transported to the Golgi complex, of course, we must now know the function of the next member of the endomembrane system, which is primarily for the processing or modification or sorting and packaging of any content, any product of the smooth as well as rough ER. Now, morphologically, if you take a look at this particular figure, it is evident that a Golgi complex is a series of flattened disc-shaped sacs. Again, it is a series of flattened sacs or disc-shaped sacs stacked together as interconnected membranes. So each Golgi stack is known as a cisternae. So this is one cisternae or Golgi sac. Behind it is another cisternae or another Golgi stack, named according to their orientation in the cell. So please keep in mind that each Golgi stack has two distinct faces. So say for instance, in this cisternae or in this Golgi sac, this face or this side facing the ER, be it the smooth or the rough ER, is known as the cis face or the cis Golgi network or the receiving face, the receiving side. And the opposite membrane, this membrane facing towards the plasma membrane or to other Golgi or to succeeding Golgi stacks, that is the trans face or the trans Golgi network or the transporting site. So please keep in mind that any membrane facing the ER, that's the cis face, those facing the plasma membrane are the trans face. So this is a trans face, this is another trans face of a cisternae, this is another trans face of a cisternae. So they all occur per stack. Now, according to research, this suits the organelle's biological activity and overall cell function. Now, how does the Golgi complex works? Well, first, you need to understand that a transport vesicle, again coming from the ER, fuses with the cis Golgi network or with the cis face, adding membrane and content to the receiving side. And so technically, products of the ER are then being modified, and as they progress further during their transit through the whole Golgi complex, eventually they will reach the trans face or the trans Golgi network of the last Golgi stack or the shipping site, giving rise to secretory vesicles different to that of your transport vesicles as it buds off and travels to the plasma membrane to secrete the said product. So to give you an idea on how the whole membrane, endomembrane system works, you have to observe and take a look at this figure. So in here, it summarizes the whole endomembrane system. So first, you have here the rough and smooth ER, and as their products are being distributed to the transport vesicles, such vesicles will now be transported to the cis side, the receiving side, or the cis Golgi network of the Golgi complex, and as it is being packaged and modified during their transit throughout the whole Golgi complex, eventually they will bud off from the Golgi complex as secretory vesicles, which then fuses with the plasma membrane as a secretion. So from the image, it is very evident that another member of the endomembrane system, your vesicles, are of different types, named depending on their function. So first you have transport vesicles, which shuttles proteins, lipids from both the RER and SER towards the Golgi apparatus. They simply transport. Whereas for secretory vesicles, as the name suggests, they secrete the contents by fusing with the plasma membrane. Now lastly, we have an incoming vesicle, which brings substances into the cell, which are then eventually digested by another member or another part of the endomembrane system, known as your lysosome. Now, if you were to ask me what is the origin of lysosomes, well, technically both the membrane as well as content or the digestive enzymes of the lysosome, specifically acid hydrolases, are made by the rough ER and were eventually processed by the Golgi apparatus. So both the membrane and digestive enzymes originated from both the RER and the Golgi complex. Now in terms of function, based on their nickname breakdown body, their function is to reduce the contents of the incoming vesicle into simpler components. A reason why, as you can see in this particular figure, an incoming vesicle, technically in the form of a food vacuole, if fused with digestive enzymes, it will reduce the contents of this food vacuole into simpler components. But besides this, lysosomes also function to break down or to reduce into simpler components non-functional organelles. Wherein in this figure, we have a non-functional mitochondrion, which will be broken down into simpler components by the contents of the lysosome for recycling. Now, if we were to answer how lysosomes specifically break down, they provide an acidic environment first to activate the digestive enzymes, and then the digestion begins. Now, as we proceed to the last member of the endomembrane system, we have now the plasma membrane, which regulates the entry and exit of molecules in and out of the whole cell. Now, for the semi-permeable property, it will be further discussed in the recorded video lecture of cellular transport. But the thing that you need to keep in mind regarding plasma membrane is that all of its biological membrane and biological membranes of other organelles, such as that of the mitochondrion, chloroplasts, nuclear envelope, ER, Golgi apparatus, and vesicles, they have the same basic molecular organization, and that they consist of double layers of either phospholipids or glycosylglycerides. Well, in particular for the plasma membrane, they are dominantly composed of phospholipids, which is this figure in this particular slide. Now, in terms of the components of the phospholipid, of course, we have the two non-polar fatty acid chains, one saturated and linear, one unsaturated and bent or kinked, both of which components of the hydrophobic tails. And then for its counterpart, the hydrophilic head, we have two other components. We have the glycerol, and attached to it, we have the phosphate. So besides these organelles of the endomembrane system, we have those who are semi-autonomous, the mitochondrion and the chloroplasts. Well, the first thing we need to answer would be, why are they semi-autonomous? Well, originally, they were both prokaryotes. One was an aerobic prokaryote, one was a photosynthetic prokaryote, living autonomously in an autonomous living body. Autonomous in a sense that they contain their own DNA. And as stated by the endosymbiotic theory, eventually a larger cell, such as this, engulf these two smaller cells, and then through time created a symbiotic relationship between them. But if they have their own DNA, why are they semi-autonomous? Well, you have to keep in mind that there are proteins and enzymes needed by both organelles produced by the nucleus. So there are those proteins and enzymes produced by the DNA of the nucleus, hence making them somehow dependent on the whole cell itself. Hence, they are semi-autonomous. Well, morphologically, both the mitochondrion and the chloroplasts are the same with respect to their membranes, such that both are double-membraned organelles. And by double membrane, meaning they have two membranes, both have inner and outer membranes similar to your nuclear envelope. But for the mitochondria or mitochondrion, they have a smooth outer membrane and a folded inner membrane, forming projections called cristae. Now, please keep in mind that cristae is very different to the term cisternae of the Golgi complex, such that cisternae is an individual sac, whereas cristae are simply projections. Now, for the chloroplasts, besides the two membranes, we have membranous sacs known as your thylakoids, an additional membrane within the chloroplast, within the semi-autonomous organelle. Now, this in particular, this chloroplast is a type of plastid, and there are other types of plastids as well. We have chromoplasts, leucoplasts, and amyloplasts. Now, functionally, they are different since one performs photosynthesis while the other most of the processes of cellular respiration, again, not all. Other organelles include the vacuole and microbodies. So for the vacuole, it is simply a large vesicle that performs a variety of functions. Hence, you have different types of vacuole. So first, you have a contractile vacuole, primarily for locomotion, usually exhibited by the paramecium caudatum, which would be this figure. So in this figure, you have the paramecium caudatum surrounded by yeasts, and inside your paramecium, these circular white structures are your contractile vacuole. For most animal cells, multicellular organisms, by the way, they have a food vacuole, or in technical terms, phagosome, which acts for digestion and cell waste management. And lastly, for all plant cells, they have large central vacuoles here, usually for storage of water, plant pigments, poison, and other unpalatable substances. Now, despite their differences, one similarity would be the name of their single membrane, which is the tonoplast. So, unlike the mitochondrion and the chloroplast or your semi-autonomous organelles, your vacuoles only have a single membrane. And its name will always be the same all throughout the different types of vacuole. Tonoplast. Again, tonoplast. Now lastly, for microbodies, a common example would be your peroxisome, which is for the detoxification of alcohol and breakdown of lipids via beta oxidation. Now, to give you an example of a cell having large amounts of peroxisome that break down toxic molecules, we have the mammalian liver cells. So, all of our liver cells have large amounts of peroxisomes, as its main function is to break down toxic molecules. Now, in terms of the origin of peroxisomes, many scientists believe that they were produced via fission from an original or from a parent peroxisome, but its main origin is still unanswered. So in preparation for the coming quiz and the first sectional assessment, we have here a table summarizing the different cellular structures of prokaryotic and eukaryotic cells. And so that is all for this video lecture. Thank you for listening.